Good afternoon. Today we're going to talk about evolution, or is it evolution? Evolution. This is a tough one because there are more questions than answers on this one. Today the questions we're going to answer, or try to address, let's say, where did viruses come from? No idea, just speculations. Where are they going? No idea, just speculations. Well, we have ideas, but it's hard to predict. How are they getting there? That I can tell you a bit about, and I can also tell you about how would we know. So for me, it's a little unsettling to not know half of the answers that we're going to talk about, but that's evolution. The techniques of modern virology lets us get at least halfway to the answer of some of these questions. We know that as host populations multiply, there are viruses that are selected for that can infect them, and this has happened since the beginning of life. We now know that new populations emerge every day. This really wasn't something that was appreciated in the 50s or even in the 60s. We also know that viral populations can shape the way hosts evolve. They can change them extensively, so they are a big force in evolution. And of course, if you can't adapt to a virus, that is particularly lethal virus infection, then you're going to be made extinct. And this is really a theoretical issue because I don't believe we have any proof for any such extinction caused by viruses, but I'm sure it has happened over time. Now, the public, interestingly, is confronted with the consequences of evolution on a daily basis, even though a good part of the public doesn't believe in evolution. It's happening right in front of them. So new diseases are emerging, like AIDS, which emerged in the 80s, West Nile coming to the US, hepatitis C, brand new infection, Ebola, Marburg. This is evolution. So I don't understand how you cannot believe in it when it's right in front of you. Flu every year, common colds, new common cold viruses, not just the same old ones, but new ones are emerged and discovered on a regular basis. Drug resistance, as we talked about last time, this is all evolution. So evolution is happening right in front of us. And I think part of the problem is that viruses evolve faster than many people can understand. If, as an evolutionary biologist once said to me, a virus is not a cow. The point is that cows take a long time to reproduce, and when they do, they make another cow. But a virus reproduces very quickly, and it makes billions of viruses. So even when you think about how these two organisms evolved, they are very different. And the other thing is that, viruses you can't even see. So how can people believe that they are evolving or that they can have these effects on host populations? So the diversity is incredible. It's actually something I didn't appreciate until not too long ago, just how diverse the virome is. And as a consequence, we're just beginning to understand the forces, the selection forces that drive evolution. We'll talk a little bit about that today, why infections emerge. And next time we will talk about specific conditions that lead to new viruses emerging. Today we're going to talk about the nuts and bolts. The third point there is very interesting, the participation of viruses in the ecosystem. Viruses have huge roles to play in the carbon cycles, in other cycles, nitrogen, sulfur cycles as well, and the turnover of various life forms. Very interesting. We're just beginning to understand this. And what does all this mean for our own survival? Just think, if you took away all the viruses, if you could somehow do that, if you could take away all the viruses just like that, what would happen? I think that we would all perish most likely, but we don't know. Someone will model this on a computer someday. So what is viral evolution? It's simply the constant change of a viral population in the face of selection pressures. And if this viral population cannot adapt, we know now it will disappear. So viruses are engineered to be able to change and adapt. We know that they do change. We've talked about it a lot in this course. We'll get a little bit at the nuts and bolts today. Diversity is amazing. If you learn one thing in this course, it is that they display incredible diversity, and that's the main reason why viruses are so successful. So we don't have great diversity. We do other things to be successful, but viruses, the key is to make a lot of viruses, and eventually one or two of those will succeed. So today we're going to talk about the sources of this diversity, which is mutation and recombination, and these are things you already know about. So much of this should be a review for you. So when we talk about populations, I also want you to know that we're talking always about, when we talk about evolution, we're always talking about populations, not individual virus particles. This will become more clear in a moment as when we define a few terms. So not the individual particles. Populations are arrays of mutants. Okay, think of the virus population as a collection of mutants, and that is what evolves, not the individual particles. And again, these mutants are made in huge quantities. Don't think of an individual virion as an average, okay? We used to think this way. We used to get a sequence and we said this is influenza virus, but it's no longer the case. So we have to always think about the population as we say, we are really population biologists, even though we don't want to be them, because that's what you're measuring when you're studying virus infections, the whole population. And so this famous biologist, Stephen Jay Gould, said, when it comes to viral evolution, the median is not the message. I don't know if any of you know who Marshall McLuhan is. Do they still teach him? Anybody know who Marshall McLuhan is? Raise your hand. No? It's amazing. Marshall McLuhan was a guy who analyzed media back in the 60s. And his famous saying was, the medium is the message. So he said if you watch television, it's not so much the content on TV, but it's actually the TV itself that gives you the experience. And so Stephen Jay Gould made a clever takeoff on that. The median is not the message. The median of a viral population is not what's important. It's not the average. It's the entire population. Because it's the population that provides the avenues for survival. It could be any of the many virus particles in a yield. It could be the survivor. And even the most rare genotype may be the most common after a single selection event. And often you don't see this. And even if we try and measure it, we don't see it in the laboratory. But this is happening all the time. So when you think about viruses, don't think about the average. Think about the entire population and what's possible from that. Now, virus evolution has four main drivers. And you could almost see this as a multiple choice question with a fifth one, right? All of the above. Large numbers of progeny, very important for evolution. Large numbers of mutants, quasispecies effect, we'll talk about that, and selection. So these are the four drivers of virus evolution. And you can see that they're very different from, say, how we evolve. We undergo selection. It takes ages to select variants of human populations. We're not quasi-species as far as we know. We don't make large numbers of mutants, and we don't make large numbers of progeny. So this is why viruses are not cows. They are a unique entity in terms of evolution. And the making of large numbers of progeny shouldn't come as a surprise to you. We've talked about this in terms of human immunodeficiency virus. These are just some numbers for HIV and hepatitis B virus to underscore that viruses make a lot of particles when they infect cells. Here is half-life of Hep B and HIV in the plasma. So HIV, an amazingly short half-life, in part a result of immune responses, but also a simple turnover. Hep B, 24 hours, daily turnover. Half of all the virions made of Hep B are turned over in a day, 90% for HIV. Total production in the blood, over 10 to the 11th for Hep B and over 10 to the 9th for HIV. So that's what we mean by huge numbers of progeny. And then similar numbers in virus-infected cells, but quantitatively different. And this is what we mean by having grounds for selection. You make a lot of particles and you have some kind of selection, whether it be the immune response, which is responsible for a lot of the turnover, or simply going into a new cell or going into a different host. So replicating viruses make a lot of mutant genomes, and evolution will happen only when mutations occur. Remember that. If you don't mutate, you can't change. And how do you make mutations? Of course, you know the answer, and that happens during nucleic acid synthesis, which is an inherently error-prone process and probably has evolved to be that way. Many experiments have been done which show that if you reduce the error frequency of polymerases, that those, whatever organism it is you're studying, don't fare well under selective conditions. And we know that for RNA viruses, these are the masters of error-prone replication. The polymerase cannot correct the errors that they make. All polymerases, of course, make errors, but the RNA polymerases don't have error correction. That's really important to remember. And as we know, the average error frequency for an RNA virus is anywhere from 1 in 10,000 or 100,000 mistakes. So one mistake in every 10,000 or 100,000 nucleotides polymerized. Right? Depends on the virus. There are variations. So again, what that means is in a 10,000 nucleotide RNA virus genome, you have one mutation per replication cycle if the mutation frequency is 1 in 10,000. So every time that genome is copied, there will be one mistake if that is the error frequency. So that's a lot, because in a single cell, the genome is copied tens of thousands of times, and that's just in one cell. And this has led to what we call the quasi-species concept. And this arose in studies of a virus of bacteria, a bacteriophage called Q beta. And it was a study which I remember. I was a graduate student at the time. I had no idea what this meant. Here's the paper. It was published in Cell in 1978. And the lead author was Esteban Domingo. This is the fellow who told me once that viruses are not cows. He said also they're not fruit flies. They're totally different from everything. A Q beta phage population is in a dynamic equilibrium with viral mutants arising at a high rate and being strongly selected against on the other. The genome of Q beta cannot be described as a defined unique structure, but rather as a weighted average of a large number of different individual sequences. So this was really never appreciated for any organism. So it came as a surprise. The reason why it was discovered is because techniques were developed that could be used to say, there's a lot of variation in this genome. Sequencing the genome is not enough to give you a sense of the variation. So this was ahead of its time, not appreciated by most virologists. So I was in good company. A lot of people didn't understand it. It took many years for me to get it. And I think even today, a lot of virologists don't understand what it means to have a quasi-species. So again, the virus populations are dynamic distributions, which means they're always changing, of non-identical but related replicons. In other words, the influenza virus replicating in a cell, the sequence is always an influenza virus sequence, but there's a huge variation in what is going on. The number of progeny produced, or I shouldn't say the number of different sequences among the progeny is huge. They all would be recognizable as influenza virus if you sequence them all, but they're incredibly variant. And that's what we mean by a quasi-species. A species had come to evolve to mean that an organism that can reproduce with other members of the same organism, and the idea was that the sequence was fixed within a certain range. And so this went way beyond that, and hence it was called a quasi-species. Okay? Now this is a picture of a quasi-species as best we can do it. We haven't been yet able to sequence an entire virus population and say this is the quasi-species, because the error rate of sequencing technology is still too high to be able to do this. We'll get there in the next few years, I think. So this is the best we can do. This is a part of an RNA virus population. Obviously, it's not the entire population because there are less than 100 members here. But you can see each of them is the genome, and it shows you that every genome is unique. in this population has different changes with respect to another, and that's what the different symbols mean. If you take an average of this population, you get the virus sequence on the bottom, and what's important is that that looks like very few members of the entire population. So the average isn't really making up the entire population. If you sequence this genome, this is the sequence at the bottom that you will get, because sequencing techniques aren't sensitive enough to pick up beyond a certain amount of change. Now, when I was doing sequencing in the 70s and 80s, you could not detect anything that was less than 15% of a population. So you always got an average, which was this here. We can now do better, but as I said, we can't get down to individual molecules yet, although we will get there. Now, because of this fact that there is a quasi-species in RNA viruses, maybe DNA viruses as well, but hasn't been as well studied, when you infect a cell or when you get infected, you're not infected by a single virion or a single sequence. You're infected by a population, really diverse population. So right away you can imagine strange things are going to happen. You're going to have all kinds of selections, and what comes out may be totally different from what goes in you. Remember, the virus that infects you wasn't made in you. You apply different selections than the person who gave you that virus. So there's going to be enormous changes. So all the progeny virions that are made in any particular cell or host are products of selective forces inside the host. So again, when you get infected with someone else's virus, the virus you get is a product of what happened in that host, and every host is different. And someday we will be able to do these experiments where we do an infection of a host and do the total sequence of what is in that host, total sequence of what's released, and the total sequence of what's made in the next host, and so on. We can't do that yet. The survivors that reinfect a new host are also the product of selective forces outside the host. So the drying that can happen, the UV radiation, the low pH, whatever the environment is when those virions are released from the first person, those apply their selection as well to the quasi-species, to the entire population. Now in the laboratory, we generally work with pretty restricted quasi-species. They're restricted because the nature of experiments are such that the cells are relatively homogeneous and so forth. And so in laboratories, you can have extreme fluctuations in genotype and phenotype because you can have selections on small populations. And as you will see, that results in massive changes. This doesn't happen in hosts, we think, because there are more diverse conditions when you go from host to host, for example. You have a lot more variability in the kinds of viruses that are produced. So here is a genome sequence. Does anybody know what virus it is? There's no way you would know. I know because it's polio, because the first 4 bases are TTTTG. But this is a myth. Now, I'm not going to ask you that, don't worry. It's just when you work with something for 30 years, you get to know the sequence. But do I really know the sequence? I don't, because it's a quasi-species, and this is just the average. When you make the RNA and you sequence it by whatever technique, you get an average, unless you go very deep, and that's what we call deep sequencing these days, when you get close to individual molecules, but not quite. Then you can pick up variations. But the sequences you see in GenBank, which are typical of this, you go onto GenBank and you search for a viral sequence, you will get a printout. And it's a little misleading. It's authoritative. This is the sequence of this influenza virus isolated from this person in this city on this date. But it's not. The virus looked nothing like this. And that's what I want you to remember, that the sequence is just some kind of a representation that we use as a starting point. So the sequence, again, clusters around an average, which is what you get when you use your techniques. But every genome can be different from every other. So it's really interesting when you think of it. You have a sequence like this, and let's say you clone it. And when you clone an RNA genome as a DNA, you restrict the sequence. You no longer have a quasi-species. You really do, in that DNA molecule, have one sequence. Maybe you have a little variation because even as a plasmid replicates in bacteria, there are some errors, but nothing like the quasi-species. So when you make mutations in genomes, starting at a DNA level, it's totally different from what is going on in the cell. So the effects you see may be totally irrelevant compared with what would happen in a normal infection. So this is something that's very difficult to deal with because many people make a living making mutations and drawing conclusions from them. But in fact, many of them may not be correct because they don't represent what happens in a population in an infected cell. So this sequence, as I said, it's very unlikely that a genome with this sequence is actually replicating. In the picture we showed before, there was one consensus sequence, but it may not be there. More likely that there are other sequences as well. So what does all of this mean in terms of evolution? First of all, it means survival of the fittest. A very rare genome in that mixture, in that quasi-species, may happen to survive a selection event, just randomly, and then you will find that mutation in progeny genomes. So it may be one genome in the original quasi-species, but if it's selected for positively, it will be found. You also say survival of the survivors, because even though that original mutation was selected for, all the others weren't selected for, they just come along with it. They are linked to it in the genome, but they're not selected because their presence is not necessary. So selection gives you a new diverse population. The only thing common with the previous population is that this one mutation or a few mutations were present in the previous population. Now those have been selected for and they predominate. But again, you have a quasi-species again. You have a highly heterogeneous population of viruses. Now DNA viruses, remember, don't have anywhere near as much variation as RNA viruses. Consequently, they have a narrower host range. I'm glad Dr. Silverstein isn't here today, who would probably argue, but he would be just being defensive for his chosen viruses because, in fact, they do have a narrower host range. Most viral infections in the world are carried out by RNA viruses. They often cause persistent infections, and maybe that's an adaptation to having a lower mutation rate because they become part of the host and may not even replicate for long periods of time. They have less replication than RNA viruses in general. Fewer genome copies are made. And of course, the genome replication is not as error prone. They have proofreading, so they can correct their mistakes. But they don't proofread every mistake. They do let some slip through. There is an error rate. It's higher. It's much lower than with RNA viruses, but there is still error. Otherwise, you would not be able to survive. A virus has to make mistakes. Any organism has to make errors in order to survive selection. So as a consequence of this higher fidelity, if you will, most DNA viruses generate less diversity and evolve slower. But they do evolve, and we have lots of evidence for that in terms of sequences. Okay, selection is the key here also, one of the drivers of evolution. This is what gives you diversity and single mutations that are selected for. So it's a little counterintuitive. In other words, you select for mutations that give you some kind of advantage. Yet, if you don't have a population to begin with, you will eventually die out. So you have to have a very diverse population from which to select specific mutations that have an advantageous effect in a certain circumstance. There are some nice examples of selection inside of hosts that we've talked about, and one of them is during the end stages of AIDS. All the virions that are present at that point are T-cell tropic. In other words, they use the CXCR4 co-receptor. Remember, HIV can utilize one of two co-receptors. So at the end stage of AIDS, the viruses that predominate, that have been selected for during the infection, which can be many years, as you remember, engage this receptor. But when you pass the infection to a new host, in that host, the initial replicating genomes are M-tropic viruses, macrophage tropic, that engage the other co-receptor, CCR5. A really nice example of selection for survival in a host. For some reason, these viruses are good at being transmitted, and these are good at replicating extensively within the host. So what we say is that these genomes have passed through a bottleneck, and only a few of the many, many genomes that are present have passed on to the new host. So the virions that eventually cause the immunosuppression are in fact not the ones that are the best to travel from host to host, which is shown by these first two bullet points here. All right, so that's an example of what I mean by selection from a very large population. Yeah, earlier I said that you can make various organisms with a lower error rate, and they don't do well. And so in fact, increased mutation rates are selected for during evolution. So not only do we select for mutants, but we select for the ability to make them. It's very important. So you can engineer mutations in viral polymerases, for example, and these changes can reduce the incorporation of errors. These we call anti-mutators. It's also been done in organisms like E. coli. And what's learned from these studies of these mutants, they don't have a selective advantage when you mix together, say, a wild-type virus and a virus with an anti-mutator together, and you ask in a cell culture which one wins, in an animal which one wins. These viruses with increased fidelity just don't do well. So lower rates are neither advantageous nor selected for. Those mutated viruses are never selected for in any experiments. And often these viruses are less pathogenic. There's an excellent example in polio, a single amino acid change in the viral RNA polymerase, which makes it make fewer errors. That mutation makes the virus less pathogenic, and it cannot compete with wild-type virus. So increased mutations rates have been selected for and are a positive force in virus evolution. So many people think of mutation as a bad thing, but what I want you to understand is that for viruses, which we're talking about today, they are essential. You have to make mutations, otherwise you can't survive. And of course, in all other organisms, it's the same, just that the rate is much lower. Now, the amount of mutation that an organism or a virus can sustain or withstand is called the error threshold. The capacity to make a lot of mutants is an advantage, as we have just told, but there is a point where you have selection and survival balanced. All right, so the fidelity and the mutation rate have to be in balance with one another. And in fact, that mutation in polio polymerase that I just talked about was selected as a consequence of this error threshold. I'll tell you about that in a moment. So the limit is called the error threshold. It's the point beyond which you cannot go in terms of the frequency of mutations. If you go beyond it, you are dead. If you live too far below it, you might not be able to survive because you don't produce enough variability. So you have to be at or near the error threshold, but you can't be above it because that means you have too many mutations in your genome and you won't make viable viruses. So remember, in a genome of 10,000 bases, you can only sustain a certain number of mutations. Otherwise, the virus will not be viable. That number is the error threshold. So RNA viruses evolve very close to it. I'm going to show you some data for that in a minute. And DNA viruses do not. They evolve far below their error threshold. So that's why they're a lot less successful than RNA viruses. So here's an example of an experiment using a DNA virus. We infect cells, and then we infect cells with a DNA virus, and then we add a base analog, which in this case is 5-azacitidine. And this is incorporated as a C into DNA. It's a modified C residue. But when it's time to copy it, it looks like a T to the polymerase, not a C. So the polymerase puts in an A instead of a G. It causes the polymerase to make more mistakes than it normally would. All right? So the structure of this base is such that it's recognized as a T when it comes to templating. When you do this experiment, the mutation rate increases by several orders of magnitude, 10, 100, 1,000-fold, for a DNA virus. So in other words, by adding this analog, you can really increase the mutation rate. But you do this with an RNA virus, the error frequency only increases 2 or 3-fold as compared to 2 or 3 orders of magnitude. So these viruses can't make any more mutations. Well, they probably do, but they're dead. So adding the drug does result in incorporation of A's in many, many genomes, but they're dead because they've already got so many mutations. They're right at the error threshold and they can't go over it. Now, there is a drug that is used to combat many virus infections. It's called ribavirin. It's a rather broad spectrum antiviral. And for certain viruses, it's been shown what this drug does is to push the virus over the error threshold. Okay, that's why it has antiviral effects. And in fact, people are wondering whether we can use this property as an antiviral to design compounds that push the virus over the error threshold. So remember, most of the antivirals we talked about last time inhibit a process, right? Receptor binding or polymerization, whatever. But this would not inhibit a process. It would make the errors go at a higher frequency and it would basically inactivate the virus. So ribavirin for polio is a mutagen by pushing polio over the error threshold. You can select for mutants that are resistant to ribavirin. Okay? You can select for mutants, viral mutants resistant of polio that are resistant to ribavirin. What do you think is the effect of those mutations that make the viruses resistant to ribavirin? Remember, the drug pushes the virus over the error threshold. Any idea? The mutations increase the fidelity of the polymerase. So the mutation is, one of the mutations is a single amino acid change in the viral polymerase that makes it less error prone. So it lowers the error threshold, or it moves the error threshold so that the virus can now accommodate what the drug does. So the drug can no longer push it over the error threshold. I guess it's raised the error threshold. So the virus lives at the error threshold initially. The drug pushes it over and kills it. But now this mutation raises the bar so the drug can't push it over. That's why it's resistant. And in fact, that is the mutant that I was just telling you about that has been used to show that viruses with more faithful polymerases cannot compete. All right, I mentioned a genetic bottleneck before, so let's talk a little bit about that. When you put selection pressures on small populations, this results in the loss of diversity and the accumulation of non-selected mutations. This often happens in a laboratory when your viruses are not as diverse as they are in a host. So for example, to do this kind of experiment, we take a single RNA virus, we do a plaque assay, we pick a plaque, and we grow up the virus from that plaque, and then we plaque it again and pick another plaque, and we keep doing this over and over again. So here's a schematic of that. So you plaque out your virus, you pick a single plaque, and then you could just directly plaque it again and so forth. You keep doing this. Right, so these are very restricted populations because there are only a few thousand viruses in each plaque. So the Quasispecies is really restricted. So what you get is a bottleneck because you restrict the virus to the progeny of a single plaque, and it's not enough variation in that plaque to make the virus healthy. So a few thousand progeny viruses derive from a single virus, the initial virus that you plaqued. So here's a little illustration of that. Parent population of viruses, you have a bottleneck, you only get a few surviving individuals, and those go on to seed the next generation. This would be better if there were 1,000 different colored balls in here, right? And then you would shake out a few colors, and then those colors would predominate. But this was written for non-viral situations, so it's not as good. So that's what is a genetic bottleneck. You are restricting the diversity of the population by these plaque-to-plaque passages. So what happens in practicality is if you do this 20 to 30 times, you will end up with a virus that can barely grow because the genetic variation is so low that these viruses are not able to propagate under different conditions. So they're markedly less fit in the original population. Now, the environment is constant. You use the same cells, you do plaque-to-plaque passage, same temperature, same virus. All you do is impose the ability to plaque from one cell monolayer to another. So what happened here? Why does fitness go away? So the answer is Muller's ratchet, which states, and this was devised before viruses were studied in this way, that small asexual populations decline in fitness over time if the mutational rate is high. So viruses, of course, have a high mutation rate. They reproduce asexually. And here's the key there, small populations. We don't have a full-blown quasi-species, and as a consequence, we have a bottleneck. So as we've already discussed, replicating RNA viruses make quasispecies. They exist at the error threshold, and that's important for survival. If we restrict the growth of this particular experiment to serial founders, the bottleneck, so many mutations accumulate that fitness decreases. So there's very little selection in this plaque-to-plaque passage. It's the same thing over and over again. You have a limited population, and you get a lot of mutations accumulating that aren't selected for, and eventually they kill the virus, more or less, because there isn't a huge pool of quasispecies from which to draw a surviving virus. So it's a ratchet because of this mechanical metaphor. Each mutation works like a ratchet, which allows the gear to move forward, not backward. So this is a ratchet. You turn this knob, you push the gear forward. It can't go back because it has this lock here. So each round of error-prone replication works like a ratchet and adds mutation at every replication cycle. And you can't remove the deleterious ones because you have a very restricted population of viruses. So this is what we've done in that experiment. So here we start out with an initial population of genomes. If you do a plaque purification, say, you now restrict the new viruses that grow up to something that looks like this. They will all have these three changes, and they will have a few others. But if you keep applying the bottleneck, which in this case is the plaque-to-plaque passage, you will have accumulations of mutations, but not a big enough pool of viruses to correct those mutations. So as a result, you lose infectivity. So this may seem counterintuitive to you, but in practice it works. We have seen this happen in the laboratory. Here are some examples of fitness decline due to bottleneck passages of different viruses. Here's a bacteriophage, 40 passages, a 22% decrease in fitness. Same with many other viruses as well. Vesicular stomatitis virus, after 20 passages, decrease in fitness. So you have to keep this in mind if you're doing a lot of passages, which you may want to do to adapt viruses to different kinds of cells. But at some point, you have to relieve the bottleneck. Now, in the real world, there are often infections that impose bottlenecks. So the key would be infection by a limited virus population. And if you get infected with small droplets of a virus produced by someone else, that's a bottleneck because there's not many viruses in that droplet. When latent viruses are activated from a limited population of cells, that's also similar to a bottleneck. Or when an insect bites you, you're getting a small amount of virus. All of these are similar bottlenecks. But these are not subjected to Muller's ratchet. In nature, viruses do very well. They make quasispecies in animals. So why is that? And how do you avoid the ratchet? Well, you subject a more diverse population to serial passage, right? You don't pick if you're doing this in the laboratory, don't pick just one plaque. Pick 10 or 20 or 30 and pool them if you need to do this. And that way you have more diversity. So why is more diversity good in this situation? Why is, if you pick 20 or 30 plaques and pool them and passage them, why would that be better? Because when you have more diversity, you can construct a mutation-free genome by either recombination or reassortment, and that gets rid of the bad mutations and substitutes for ones that allow viral growth. Okay? So the simple fact of adding a greater diversity in this experiment allows you to escape the ratchet. So you just pick more plaques instead of one. So you may think that how likely is it that recombination would restore the fitness? Well, you only need one viral genome in the entire population that's fit, and it will overgrow all the others and predominate. So if that one genome is produced by recombination just randomly between two viruses and it happens to produce a genome that is now fit for whatever reason, that will take over. So even a very rare event will win. The progeny will take over the population. So all of this means diversity is important for the survival of individual members. So remember, we're talking about virus infections on a population basis, but the population is important for individual viruses because what comes out is really an individual genome that goes on to the next cell. So if you take away diversity, the whole population suffers. So this is a slide that just shows you how reassortment or recombination in this case would give you a healthy recombinant. Here are two viruses that have been produced during this plaque-to-plaque passage. They have lethal mutations in their genomes, so they cannot propagate. But if you recombine the two genomes, you now have a healthy viral recombinant. As I said, even if this is very rare, that one virus will survive. All the other ones won't be able to. This one will, and it will replicate. So the exchange of genetic information is really important in the viral world. We haven't really talked about it much in this course. We have talked about it in terms of doing experiments, but it's incredibly important to allow viruses to escape these sorts of ratchets that restrict diversity. Now, RNA viruses that have segments like influenza virus and reoviruses, they do a similar thing. They don't recombine, they just reassort. The segments are mixed. So it's the same principle. That kind of reassortment can rescue the lethal genomes. And it's an important source of variation. We've talked about how reassortment is important to generate new influenza virus strains. It's the same for reoviruses as well. So that brings us to different kinds of selection that occur. We've talked about selection in general, but I want to talk about two specific kinds that we see in virus populations: genetic shift and drift. When viruses replicate in an individual that makes a robust immune response, or a so-called immunocompetent individual, viruses will arise that can evade the immune response, whether it's a T-cell response or an antibody response. Remember, you have a diverse viral population. Among those members, there's going to be one that can escape a particular antibody or a T-cell, and this happens all the time. So there are two kinds of such immune escape. There is drift, and this is diversity that arises from copying errors followed by immune selection. So this can happen every time a genome replicates, and this happens for influenza virus. And then there is shift, which is the kind of diversity that you get after recombination or reassortment. As we've talked about before, this is relatively rare, but it doesn't matter because the survival will predominate. So both of these happen very prominently with influenza virus. So we've talked a little bit about the pathogenesis of influenza and the acute nature of infection. But let's go a little bit into drift and shift. And to do that, we have to understand a little bit about the antigenic composition of these viruses. You remember there are two major glycoproteins in the virion of influenza, the hemagglutinin, the HA, and the neuraminidase, the NA. So here we have HA and NA. And because there's such extensive drift and shift among influenza viruses, a system for nomenclature has evolved where you have to give a number to the type of HA and the type of NA that's on the particular virus. So it's H, X, and Y. So far we know there are 15 different kinds of hemagglutinins and 9 different neuraminidases, so you can have different combinations of these in various animals. Now, the H1, 2, and 3 hemagglutinins are the ones that can infect us. Viruses with these can infect us. And all of them can infect birds as well. Okay? These H's, the H1, 2, 3, 4, all the way to 15, have all arisen by drift over the years. And then reassortment of these different H's and N's gives us new pandemic influenza viruses every 20 or 30 or 40 years. And that's what you see in this slide. Let's start in 1918, for which we have a virus. This is the first outbreak or pandemic of influenza where we actually have the virus because we reconstructed it. We don't have any viruses for the pandemics of 1900 or 1889. But from studying the serum of people who lived them, we suspect that these were the kinds of viruses that caused these pandemic, an H2N2 and an H3N8. But we're not sure. So in 1918, the pandemic was caused by the so-called Spanish influenza. It was an H1N1 strain. And by studying viruses that circulated in ducks, it looks like this is an avian virus that somehow jumped into humans. So all the RNAs are red to show that it came from this red duck. Okay? Now, this virus circulated for many years until 1957. It caused yearly outbreaks of influenza because every year or two the HA would change, 1 or 2 amino acid changes imposed by genetic drift and selection by antibodies in the hosts. But then in 1957, there was a major pandemic of a totally different strain, an H2N2 strain. So this is not recognized by any of the population. What happened was the H1N1 reassorted with a duck H2N2, and the yellow genes came from the duck. So this is now a random reassortment that happened somewhere. We don't know what species it occurred in. And then that virus somehow got into people, and we didn't have antibodies to them because the H and the N were totally different. So it caused a global epidemic or a pandemic. 1968, so that virus circulated from 57 to 68. Again, in 1968, this virus reassorted with another duck virus, the blue genes. You can see a couple of blue genes. And now this is an H3N2. So it has a brand new hemagglutinin, same neuraminidase as the previous virus, but that was enough to cause a pandemic. Same thing happened in 1977. Now, just in 2009, again, we had a pandemic of influenza virus, the 2009 H1N1, which was a swine-derived virus. And this virus, again, was derived by reassortment in animals. It had 4 different parents. It had one parent was a virus that infected Eurasian swine. These are pigs raised for the pig food market, both in Europe and Asia. There's an extensive trade between those two countries. So that has all these green segments, and you see 2 of those ended up in the H1N1 of 2009. Then a classic swine H1N1. This is viruses that have been circulating in pigs since 1918. A human H3N2, and a bird virus. So all of these reassorted over a 10-year period, probably in pigs, to form 2 intermediate viruses, which then reassorted to form this. And this had all the right sequences to infect people. If people weren't around, it would never have infected them. But of course, we make the pig farms, so it's all connected. In fact, the pandemic flu seems to be have become more serious since the emergence of pig farming. Pigs serve as a nice vessel for all these strains to replicate in. Anyway, it's an example of how reassortment probably provided some advantage to this virus, say in a pig, but just by chance, that virus happened to do well in people. So that's antigenic shift, major changes by reassortment or recombination. Drift is, again, minor change in flu. It happens from year to year as the virus mutants accumulate. They're selected by the antibody response in populations. And eventually you have viruses with single amino acid changes in the hemagglutinin right at the tip. And these are near sites where antibodies usually bind to neutralize infectivity. And so this is why we have to change the vaccine every few years, because you have a gradual accumulation of mutations. Again, it's selected by our antibody response. The virus can tolerate these changes, so it slowly escapes the antibody response, and you have to make a new vaccine because the old one doesn't work anymore. So that's antigenic drift. Some other examples of exchange of genetic information, we've talked about reassortment as a means of exchange. There's also ways in which you put non-viral nucleic acid information into viral genomes. We have talked about most of these so far in this course. Dr. Silverstein talked about recombination leading to the production of cytopathic bovine viral diarrhea virus in cows, the production of oncogenic retroviruses. Remember, picking up oncogenes from the host cell by RNA tumor viruses. This is an example of exchange of genetic information. And viruses that have picked up virulence genes related to host immune defense genes. The viruses have homologs of cytokines or cytokine receptors. At some point in evolution, as those came from the host cell, there was some selection imposed on those viruses that randomly acquired those. So this is just a diagram of the bovine viral diarrhea recombination event. This virus here, the genome of this virus, an RNA virus, this virus is non-cytopathic in cows. But at a very low frequency, it picks up a piece of RNA from the host cell that encodes a ubiquitin cleavage site. So that's what UCH stands for. Ubiquitin is a protease found in host cells. So it's picked up the cleavage site. There are 2 different kinds of viruses, one with the cleavage site put here and another here. The result is now these proteins can be cleaved, whereas previously they are not, by cellular proteases, and the virus is more virulent as a result. So this is a chance recombination event that occurred during infection, but it has a powerful advantage, so it's selected for. Another example of how evolution proceeds, this one by host switching and recombination. And this involves two viruses, very interesting story. One of them are circoviruses. These are vertebrate DNA viruses, really small genomes, don't encode many proteins. And the other is a nanovirus. Same genome structure, but they infect plants. So we have circos and nanoviruses. The rep genes, now these viruses encode a protein called rep, which is not a polymerase, but it's needed for DNA replication. It's called rep. These are hybrids of the two viruses. If you look at the sequence, you can see that both viral genomes are present in these sequences. They also have homology with the RNA binding protein of a virus from a different family, the caliciviruses. The hypothesis, having looked at all this sequence information, is that the nanovirus, which infects plants, was at some point long ago transferred to a vertebrate. Maybe a vertebrate was drinking sap from a plant and the sap had virus in it, so the plant virus somehow got a hold in the vertebrate. And that established the circovirus family. So first they were nanoviruses. The viruses jumped from plants to a vertebrate and established the circovirus family. And then after that, recombination occurred between the circovirus and a calicivirus, which gave it this homology to an RNA binding protein. So you see there are many interesting ways that recombination and reassortment give rise to new viruses. An interesting question, which we haven't really talked about in this course, is, while we're talking about selection, is virulence a positive or negative trait of selection? Is this something that is selected for or against? We're all familiar with virulent viruses that devastate populations, but is that good for a virus to be so virulent? And one of the considerations is that increased virulence reduces transmissibility, because if the host dies too quickly, it can't transmit the infection. So this is one argument why evolving towards increased virulence really isn't a good idea. So if you accept that increased virulence isn't good, you would think that all viruses evolved to be maximally infectious or transmissible and avirulent, right? Why should there be any virulent viruses at all if they have no advantage to the virus? But we don't actually observe this. There are lots of viruses that are quite virulent, so there must be a reason for that. And one idea is that perhaps virulence increases transmissibility. So if a virus causes you to cough and sneeze a lot, which is an increase in virulence, that will help it to transmit. We see also persistent infections that are rather dormant for years, so they're not virulent, but then at the end of the infection they kill the host. So there must be some advantage to that kind of viral life cycle. We also see cases where there are viruses that are quite virulent for one species, but in another animal they're asymptomatic. So here, the same virus in two different species has a very different virulence phenotype. So we're not quite sure what the relationship is between selection of a virulence as a positive or negative trait. As I said earlier, maybe increased virulence increases transmissibility, and I think transmissibility is probably the most important selection factor, so maybe virulence sometimes comes along. There's no answer to that, of course, but those are some interesting points. Now I want to tell you about an experiment in evolution that addresses this virulence and selection issue. This was done in Australia, and in 1859 the Australians brought in some European rabbits so they could hunt them. They like to hunt rabbits, so they brought in these rabbits and they multiplied, but unfortunately there were no predators of these rabbits, so they got out of control. There were just millions and millions of them, and this is one photograph from Australia showing how many of these guys there were. So the Australians got upset. They said, we have to do something about these rabbits. So what they did is release a virus, a pox virus called myxomavirus. They said this virus is quite lethal for these European rabbits. So the natural host of this virus is from California and Central and South America. In those rabbits, the virus infection is not very harmful. It is spread by mosquitoes. In the European rabbits, though, which is not their natural host, the virus is 90 to 90% fatal. So that's why the Australians decided to bring myxoma in and release it, and mosquitoes would pick it up and spread it among these rabbits and get rid of them. Okay, so how did that work out? First year, the virus killed 99.8% of the rabbits, but the 0.2% who survived, they multiplied like rabbits. The second year, mortality dropped to 25%, and then the rate of killing and subsequently ended up being lower than the reproductive rate, so the rabbits are still in Australia to this day. This is actually a topic that's been extensively reported on in the press by a number of virologists. So you can go back and look at all this old literature. So it's an interesting example of coevolution of the host and the virus. The virus doesn't want to kill all its host as far as viruses can want anything. So it evolved, and the host also evolved to be more resistant. So there are changes in the rabbits. You select for the survivors that can resist infection. There were changes in the viruses so they wouldn't wipe out all of the hosts as well. So this is interesting. Both rabbits and viruses produce large numbers of offspring. This is probably why this could be observed in such a short period. So the virus evolved, as I've said, to kill fewer rabbits and then extend the life so the virus could be spread in mosquitoes. So those viruses present in the population that had this property, just randomly, it was one of the quasi-species in the population, were selected for, and those were the ones that predominated. And this is exactly what I would predict, an evolving host coming to equilibrium with the pathogen. So the European rabbit was a new host for this virus. So this is an evolving relationship. So both the host and the virus evolved so that both can satisfy their needs. So what was learned from this? You always get what you select, but you often don't get what you want. Okay, so they wanted to wipe out all the rabbits, but they didn't. But they instead imposed a selection which allowed both to survive. So this is a really bad idea to try and eliminate any animal with a virus. However, the Australians have not learned because experiments are still ongoing to try and get rid of these rabbits using other viruses. There's a calicivirus. The caliciviruses are RNA viruses, and they're trying to exterminate the rabbits with them. And last year when I taught this course, there was just an article in the paper where this virus had accidentally gone out of the containment station where they were doing limited experiments on rabbits. So another accident happening in Australia. So the European rabbits are still there, and the moral is that you really can't get rid of them with this approach because the evolution will go its own way. All right, where did viruses come from? As I said, we don't know, but we have some theories, and they're worth discussing. There's the regressive theory, which says that viruses are derived from intracellular parasites that have lost most of their genes. There's the cellular origin theory, says that viruses arose from cellular components that somehow got the ability to replicate. And there is the independent entity theory. I didn't make up these names. These are terrible names. Independent entity. Viruses co-evolved with cells from the origin of life. I actually like this one. I think that viruses were there before cellular life evolved. Now you may say, how is that possible? Because viruses need cells, but it's our definition today of a virus. It may have been they existed in a very different form before cellular life evolved. There is a little bit of evidence for a few of these when you look at the evolution of viral genes, which we'll talk about in a moment, but obviously we can't prove any of it. So we don't have any fossil record, not like we have of vertebrates. We do have genetic fossils. We have viruses and genomes of animals, and we can tell by that how many millions of years ago those viruses were around. But we don't have old virus stocks. It's very hard to test hypotheses. But we do study contemporary viruses and get some ideas. And so here are some ideas from that about the origins of viruses. First of all, there are viruses of all three domains of life, the bacteria, the eukaryotes, the archaea, and they all have homologous features. They have things that make them look like viruses. And this suggests that the viruses predate the last universal cellular ancestor, also called LUCA, which is presumed to be ancestral to the three domains of life. So maybe viruses were there before these three branched out. The DNA viruses of green algae may be the oldest ones. Their genomes are what we say at the root of most eukaryotic sequences. They are ancestral to everything we know of in the eukaryotic world. So we think these are probably the oldest eukaryotic viruses. And as we sequence more and more viral genomes, we find more genes that have no identity with any of the genes in us or in any other member of the three kingdoms. So that makes it hard to say that viruses came from something living because they have genes that aren't in any of these things. So they're more likely to be around before them. So here's an example of this. Some of these very large viral DNA genomes that have been discovered lately. The Mimi viruses, 1.2 million base pairs, Phycodnaviruses. These are viruses of algae, the ones that we think are at the root of eukaryotic sequences. These have sequence coherence. That means they're not mosaics. They are not mixtures of genes from other viruses. All the genes they have are unique to those viruses. They haven't come from anywhere else. And that is difficult to explain using a model that they arose by getting genes from another precursor genome. So we think that these are very, very old viruses. They happen to be among the largest, and that may be why they are so big. Maybe they were even bigger at one time and replicated autonomously and then lost genes and became viruses. These mimes, by the way, infect amoeba. We now know over 1,600 viral genomes, probably even more today. We can put all those sequences on a glass chip, and it's called a virochip, and then we can use that to probe environments for the presence of some of those viruses. We don't have to grow all the viruses anymore. This has been a really powerful development. We can sample viral diversity without having to grow the viruses, and we've discovered many, many viruses this way that we can't grow. One project was done to purify capsids from the water. So all the viruses in the oceans have been sequenced this way. And sequence the DNA and just see what's there. And when we do this, when we do these massive metagenomic studies, we find most of the genes are unknown. We haven't seen them in any virus, and we certainly haven't seen anything like them in cells. So there's a huge amount of diversity out there that we haven't sampled, but more importantly for our discussion, it argues that these viruses evolved on their own and not from cellular forms. Here's an interesting story of the evolution of herpes viruses. Now you can sequence all the known herpes viruses and create a time scale of evolution. And when you do that, the three major groups, the alpha, beta, and gamma herpes viruses, you can calculate to have arisen 180 to 220 million years ago. So that's not very long ago in life on Earth. So here's formation of Earth 4.5 billion years ago, and the emergence of life maybe 3.5 billion years. And then here we have the dinosaurs right about here. That would be 250 million years ago. And so we're saying the herpes viruses probably arose during the time of the dinosaurs, not very long ago. But we don't have anything before that so far. Also, there are other herpes viruses that infect fish and oysters and amphibians. They have no sequence homology to these human and animal alpha, beta, and gamma herpes viruses. They have a capsid like them, and that's why they're placed in the herpes virus family. But these may represent a very early branch of the herpes family because they have no gene homology to the human herpes viruses. Smallpox, by sequencing 45 different isolates, you can put them into clades that have geographic clusterings. All of these viruses have the same number of genes that seem to do similar things. Very little diversity, which suggests that they were introduced recently into humans. And in fact, just one member has significant homology to smallpox, and that is a pox virus from gerbils. So the idea has arisen that smallpox virus arose after a zoonotic infection from infected gerbils. So zoonotic means going from animals to people. So smallpox, which devastated humanity, may have arose from gerbil a number of years ago. Papillomaviruses and polyomavirus, these small DNA-containing viruses also co-evolved with the host. And a very interesting story is that the distribution of the type 16 and 18 of these viruses is totally congruent with racial and geographic distributions of human populations. So there are specific populations globally that have certain genotypes and certain variants within each genotype. So you can actually map the movement of populations by what kind of HPVs they have. Another virus in this family, JC virus, is also a marker for migration. You can tell where people have moved from Asia to the Americas by just looking at the kind of a JC virus genome that they have in them. It's really amazing. And in this case, the infections appear to spread vertically, that is from mother to newborn, which would explain this kind of association rather than being spread by respiratory routes, for example. Measles virus is closely related to a virus of cows, rinderpest virus. And we think measles arose from rinderpest when humans first began to domesticate cattle. So we got close to the cattle as opposed to them wandering wherever it was they wandered. We started domesticating them, we're right next to them so we could pick up their virus infections. And in fact, domestication of cattle happened in the Middle East about 5,000 years ago. And then the virus, after being introduced into humans, spread around the world by colonization and migration, and eventually was brought to the Americas in the 16th century and destroyed many, many Native Americans there who had not colonized cows and hence were not exposed to it previously. Now, an important point at the end of all this is that viruses remain viruses no matter how much they evolve. Okay? Despite all the replication and selection, we can see a herpes virus, we can see an influenza virus, we just look at the genome. That's why a quasispecies, all those variants, we can still tell what kind of virus they are. So there's a lot of opportunity for variation, but you do vary within a master sequence of some kind. How is this so? Why do viruses completely change? Well, they are defined by their capsids and their genomes and the way they replicate. So we think that's a very strong restriction. They all share fundamental characteristics, and it constrains them. So any virus that can function within this constraint will survive, but a DNA virus cannot become an RNA virus, apparently. So we have now 7 different genome types and no more than that, and that's probably all we will ever see. So any extreme alterations don't survive selection, going from DNA to RNA. And of course, as I've said, the genome is one constraint, as is the replication strategy, the nature of the capsid. You can't change from an icosahedral capsid to an envelope or vice versa. And there's also selection during host infection. You can't change the way you infect the host or else you'll be eliminated. So that's what we mean when we say viruses are constrained by their structures and modes of replication. Now, new viruses, we assume, will only arise for the ones that are here right now. We're not going to have any totally new ones. Everything that comes in the future will arise from what we see. So what is the number of all possible mutations, just to get an idea of what's possible? In fact, it's so large that it can never be tested in nature. When you sequence RNA viruses, you see that over half of all the nucleotides can be changed in any given RNA virus. So let's do some numbers. For a 10 kilobase viral genome, that means there are 4 to the 5,000 sequence permutations that define all possible mutants. So we're assuming half of the genome can be substituted. So how many substitutions is that? 4 to the 5,000. And then it's probably bigger because deletions, recombination, and reassortment would add to it, but we're not taking that into consideration. But the universe only has 4 to the 135 atoms. This is a huge number. Remember, at the beginning of this course, we talked about the total number of viruses in the ocean, which 10 to the 30th, I think, which is 200 light years away if you line up all those viruses. So this is even bigger. So that's why we can't predict the future. So HIV, of course, gives you huge opportunities for selected. We do tens of millions each day in a human. You produce billions of genomes a day, and that's where the number of 10 to the 16th genomes for HIV comes from, which we ended up last lecture with. And the mutation rate is such that every genome has a mutation, and every human produces genomes with many mutations that result from mutation, recombination, and selection. So that's why we have resistance to every combination in use or in the pipeline produced by chance every day. And most of the time they aren't selected for, luckily. So virus evolution really is relentless. We will always have mutants, and the possibilities really are unimaginable. The only thing we can say is that the new viruses that arise will be mutants, recombinations, or reassortments of what is out there. Certainly we could have two different viruses recombined, but they would both have to be DNA viruses with the same genome and the same capsid, most likely. So they will resemble what we already have. We only know about 1% of the viruses that exist on the planet now, so you can imagine that we still have a lot to learn from this. And I'll leave you with this thought: as mutation rates are probabilistic and viral quasi-species are indeterminate, the concept of prediction has little meaning. So the numbers are so huge that we can't really predict where viruses are going. But let me leave you with this interesting tidbit. It took 8 million years for simian primates to change 2% of their genomes to become human beings. 8 million years. In contrast, polio can change 2% of its genetic structure in 5 days. And that is the time it takes the virus to pass from your mouth to your gut. 5 days, and it will change 2% of its genome. So that's why you see that viruses can be so successful, because they change so rapidly. So viruses are not cows, they're not humans, and they're not Drosophila. 8 million years is really nothing in terms of evolution, and it's unfortunate that we can't study that, but life has been around a lot longer, so you can imagine what viruses have done.